Hello everyone, my name is Shraddha KATARPAWAR and I'm currently pursuing my PhD from the Department of Pharmaceutical Analysis from NIPER, Hyderabad. Today, my topic of presentation is proteomic study of human cord blood reticulocyte derived exosomes. So, uh, the corresponding author in my manuscript is Dr. Hernando A. del Portillo. Uh, Dr. Hernando A. del Portillo has a great expertise and is recognized scientist in the research of exosomes and in blood-related diseases like anemia and especially in human Vivax malaria. Sir has also played an immense role in the discovery of biomarkers and in vaccine development. So, coming to the topic of exosomes, since in human cord blood means the blood which is present in the umbilical cord, which contains large number of reticulocytes, which are also called as immature RBCs. When these immature RBCs get converted into mature RBCs, they release some proteins which are called as transferrin receptors and some enzymes. So when these proteins comes in contact with the cell membrane, they get converted into the vesicle, extracellular vesicle called as exosomes. So exosomes contains majority of this transferrin receptors, and this transferrin receptor also known as CD71, which also act as a marker for exosomes. So what is the research question in this particular study? So, research question is like there are research question. What are the specific type of protein that are present in the exosomes? Then, what are the function and role of the protein in various biological processes? Then how this protein and it interfere in the biological pathways, and what is the role of this protein called as exosomes in various biological pathways, and what is the protein composition of this exosome. So to address these research questions, some objectives are designed in this study. Firstly, mass spectrometric analysis. Then next we are going to see the cell based assay, and then proteomic analysis. So in methodology, the first step is the purification of human reticulocyte. This human cord blood sample, it was collected from the hospital in Barcelona with the regulatory approval, and then this cord blood was centrifuged, and the supernatant was collected, was discarded, and the remove the deposited pellet, which we are going to wash with the RPMI 1640 media. Then what we can say, the extract which we got after removal of the supernatant and washed with the RPMI, the remaining cells, these were washed with again twice and thrice with the RPMI medium. Then leukocytes and platelets, these were removed by using filtration through the cellulose column. This reticulocytes, these are enriched by Percol, uh, uh, by the Percol solution. The Percol solution in which our sample is added, and then Percol in 70% Percol solution our sample is added. Then we are going to centrifuge the sample. Then the layer will like interface layer will form, and in the interface layer our sample which is of our interest means reticulocyte, which will get deposited in the middle of the layer. So we carefully extract it out. So hence we took the sample of like this. We are going to purify our human reticulocyte, and the next step is in vitro production and isolation of human reticulocyte derived exosomes, which can be done by two methods: ultracentrifugation and size exclusion chromatography. So the reticulocytes, these were washed with these are cultured in RPMI medium, and cell viability was checked by using a dye called as trypan blue, means the reticulocytes were going to stain with this trypan blue dye. Then exosomes are isolated by two methods, which we called as size exclusion chromatography and ultracentrifugation method. Then exosomes in supernet, this supernatant was loaded onto what we can say a film uh, made up of film or column, which is made up of cellulose, and the liquid, or we can say the sample, we are going to pass from the column. So hence the liquid, the fraction we are going to collect is called as the SEC fraction. And in ultracentrifugation, the samples were subjected to the sequential centrifugation at high RPM. So the supernatant is obtained. It is called as ultracentrifugation supernatant. So by these two processes, we are going to isolate this reticulocytes. Now both the extract are enriched with reticulocytes. The next step is the nanoparticle tracking analysis. Basically, this nanoparticle tracking analysis is performed to know the size distribution and the concentration of the reticulocyte in a particular sample. Next is bead-based flow cytometry assay. In bead-based flow cytometry assay, the sample which we got from the previous steps, like for SEC and UC, ultracentrifugation, these samples were then coupled with the aldehyde or sulfate latex bead. The bead is made up of like bead is called as the aldehyde or sulfate latex bead. So when we are going to react, when we are going to couple this our supernatant and bead, like bead work will get code, bead will interact with our sample, so that reticulocytes will get attached to the bead. And after coupling, we are going to add the bead coupling buffer. Then we are going to wash that bead, and after washing, the exosomes get the exosome coated bead. These were incubated with CD71. Yes, with CD71 primary antibody. Then again washing is done with like 2 to 3 times, and which is and then it is again incubated at room temperature, and the flow cytometry assay we're going to perform. The next step in this analysis is immunoblot analysis. In immunoblot analysis, like western blotting for protein of interest called the CD71 will be done. Then again immuno western blotting of the previously identified protein HSP70, GAPDH, and stromatin will be performing. Then next is the transmission electron microscopy. Transmission electron microscopy for ultracentrifugation is performed, and along with the immunogold labeling, exosomes and virus-like protein capture by mature by this mature, what we can say, monocyte derived dendritic cells via cyclic 1. Here cyclic 1 is a receptor which is present on the surface of the dendritic cells, and these dendritic cells are going to capture the exosome. So this capture ability will be checked by using this method. And the last step is the mass spectrometric analysis. So the new technique, and coming to the new techniques and instrumentation. So in this particular research paper, I found that immunoblot assay, which is something I have not previously heard. And in immunoblot assay, we are going to, what we are going to do is we are going to prepare the sample, and after preparation of the sample, we are going to keep the sample in the film, and we are going to incubate it with the CD71. After incubation with the CD71, this film is going to wash, and after that we incubate it with the primary CD71 antibody. And after incubation with the primary CD71 antibody, we are going to again wash it. Then again we'll couple it with the secondary anti CD71 antibody. So after this, we are going to perform the protein analysis and then ultimately data analysis. So these are the results. This is the result obtained by the obtained for the isolation and characterization of exosomes derived human cord blood reticulocyte. The first diagram which is shown for NTA profiles of human reticulocyte derived proteins from ultracentrifugation and size exclusion chromatography fraction. uh, it is shown in the particle per microliter. Since in the graph 1, we can observe that in ultracentrifugation section, the intensity like 127, it is high as compared to size exclusion chromatography, which is of 160 and 158. Its intensity is still low. It means that ultracentrifugation is an ideal method of isolation as in NTA profile. Thus, NTA profile suggests the greater intensity of ultracentrifugation method. Then next we are going to see the results of flow cytometry. In flow cytometry analysis, the transferrin receptor, which is like flow cytometry analysis, is particularly performed for detection of this CD71. Means the intensity here in graph B will represent the amount of transparent receptor which is present in a particular extract of size exclusion chromatography and ultracentrifugation. As in size exclusion chromatography, we are supposed to see the lower peaks. Means the intensity of the CD71 receptor is low in case of size exclusion chromatography, but in ultracentrifugation the intensity is very high, which we called as median fluorescence intensity. This median fluorescence intensity is nothing but it indicates the amount and what we can say the presence of CD175, the CD71 receptor. The more the intensity, the more the median fluorescence intensity, the more is the amount of that transferrin receptor. So hence we can see in case of size exclusion chromatography, the values are little less as compared to ultracentrifugation. So here also we can conclude that ultracentrifugation is a better process as compared to size exclusion chromatography for isolation and characterization. Then third image which we are seeing is electron microscopy. In electron microscopy, we are able to capture the size and diameter of the of the exosomes. So these spherical shape particles are called, these are the exosomes, and which are ranged from 200 nanometer. Next result is the detection of the transferrin receptor in human reticulocyte derived exosomes. The first result which is shown to us is immunoblot in human reticulocyte derived proteins, which is purified by ultracentrifugation method and size exclusion chromatography fractions. So this immunoblot this immunoblot assay which indicate its result in the form of bands. So higher, like greater the intensity of the band is, means significant the results are, or more amount of, like if the band is more intense, then it is what we can say is that our substance or compound of interest is very separated very well, and it indicates the intensity or the presence of that particular protein or particular compound in our sample. So in ultracentrifugation, as we can see that the dark band appear, means the transferrin receptor is detected very well in case of ultracentrifugation sample, whereas in size exclusion chromatography we can see a very lighter bands or very thin bands, since the intensity of the band is very low in size exclusion chromatography. So we can say that transferrin receptors are not much well detected in case of size exclusion chromatography extracts. So in ultracentrifugation extract, transferrin receptors are really more as compared to size exclusion chromatography. So the second image is of immunogold labeling of ultracentrifugation human reticulocyte derived exosomes using a secondary antibody conjugated with the 10 nanometer gold sphere, which scale bar represent in 200 nanometer. So this figure 3, it consists of the proteome and human reticulocyte derived exosome, which are identified by LC-MSMS. For the first graph which we are seeing is the Western blot validation for the known three proteins. HSP70, GAPDH, and stromatin. This HSP70, HSP70, GAPDH, and stromatin, these are already identified protein in the previous study. In the previous study, this out of like all only seven proteins were identified in the previous study, but in this study a total of 376 or 390 something proteins they have isolated. So this the samples in Western blot. These were purified by the two cord blood donors, each one loaded in a different lane. So as we can see, this SSP70 contains a very darker band as compared to GAPDH and stromatine. The graph B, it represents the distribution of the proteome of human reticulocyte derived exosome in subcellular location categories retrieved from Uniprot database according to gene ontology and annotation. So as we can see that majority of the protein, like 25% of the exosome, which occur in the extracellular region. 15.9% occurs in cytosol, 14.9% occurs in plasma membrane, 13.5% occurs in nucleus, 5.9% occurs in cytoskeleton, 5.7% occurs in lysosomes, 5.7% occurs in endosomes, 4.5% occurs in Golgi apparatus, 3.9% occurs in the endoplasmic reticulum, 3.4% occurs in mitochondria, and 1% in percent is still not retrieved, and 0.5% is present in peroxisomes. So this is the distribution of proteome of urex. So majority of it is located in extracellular region. So this is the Siglec-1 dependent capture of human reticulocyte derived exosomes by mature dendritic cells, of which the first figure consists of the flow cytometry analysis of the urex DIL, which is urex DIL means it is the labeled urex captured by the MDCs. So as we can see that urex DIL here value is 19.88 plus minus 8.43, means the capture is less as compared to the negative one. So as we already discussed, like these dendritic cells, these cells have the capacity to capture the exosomes, and this capture is done by the monocyte derived dendritic cells. So this figure B, it represents the confocal microscopy co-localization of the urex DIL, which is the labeled human reticulocyte derived exosomes, which is denoted in red, and this virus-like protein HIV. This is the tag called. This is the tagged protein or labeled protein called as GFP, which is encoded in green in mature like in monocyte derived dendritic cells, in which nuclei is stained with the DAPI dye. So in conclusion, this is the first mass spectrometry based method or the first mass spectrometry based proteomic analysis of urex, which provided us 367 proteins. Earlier this study only 7 proteins were isolated and quantified, but in this study that too, this is the proteomic analysis where we have got 367 proteins which are identified and quantified. Next conclusion is that thus human reticulocyte derived proteins, it can facilitate antigen presentation via dendritic cells and may play a role in red cell diseases such as sickle cell disease and malaria. And this study, it opens a new avenues for further research on the biogenesis of these exosomes and their roles in immune responses and their potential as biomarkers in the red cell diseases, since the red like they interfere in the what we can say, like these events occur between the process of erythropoiesis, which is called as the formation of the red blood cells. Cells from reticulocytes, they when they are converted to mature RBCs. So in between that, these events occur and it leads to the formation of exosomes. So there may be a chance that in diseases which are associated with RBCs, these exosomes might interfere, and how they are altering the biological process. So what I learned from this manuscript is, I have never heard of the concept of exosomes. So through this manuscript, I was able to know about what are exosomes and what are its composition and how the analysis we are doing. Then about the quantification of proteins in the exosomes. Exosome itself is such a tiny particle, and its protein content means like it's a very good opportunity to learn about the quantification of proteins in exosomes, and what are the role. And I have also learned about the role of exosomes in various biological processes, especially in erythropoiesis and in red cell diseases. Thank you.